Hi, Michelle Glass here. Welcome to another episode of our Chapter 19, Lecture 19 series. And we're still actually looking at those red blood cells or erythrocytes. And what we're going to talk about in this video is what's referred to as blood typing. So we're going to be looking at actually two different components of blood type. So we're going to be looking at what's described as ABO blood type, and then also the RH blood type, which you probably are more familiar with as like positive and negative. Before we actually start looking at blood types, we need to define a few terms. The first term that we need to define is the word antigen. Antigens are named because they generate immune responses. So what we see is that all cells have little protein markers as part of the cell membrane. Remember, we talked about glycoprotein markers way back in AMP1. Those markers are what our immune cells can use to recognize which cells are self and then which cells are foreign. So we have self-antigens, so these are going to be those markers that our body recognizes as the cells that belong to our body. And so in that case, we say, oh, that's a Michelle Glass cell, don't attack it, right? Okay, but when we see that it's a foreign antigen, then our immune system will perform some kind of immune response. Very often, this is going to lead to the generation of what are called antibodies. Antibodies are going to bind to those foreign cells and really enhance our immune system's response, primarily phagocytosis. So remember, that means cell eating. When we are looking at our blood type, our ABO blood type, we will see both the discussion of antigens and antibodies here. Now, the key thing to remember is that in ABO blood typing, both the antigen on the red blood cell and the antibody is an innate part of your genetic inheritance. So if you get the information to have type A blood, then what that means is that your cells will have on their cell membrane this antigen that is specific to type A. So here I've just been goofy, and I used my little A on the outside of my red blood cell to sort of represent that antigen. The other thing that you inherit with type A blood type is an anti-B antibody. So this means as part of your genetic inheritance, you have this fairly large antibody that's circulating, and so if you were to be exposed to type B blood, you would attack those red blood cells as foreign, just the same way you would a pathogen. And then we see pretty much the opposite for our type B individuals. So here you have the B antigen being produced and hanging off of the cell membrane. So B is recognized as self, and also as part of your genetic inheritance, you have this pretty large antibody called the anti-A antibody. So in this case, you're going to recognize any blood cells with type A antigen as being foreign cells, and you're going to attack. You're going to do an immune response. Now, if you are type AB, what this means is that you have red blood cells with both A and B antigens hanging off of them. And so obviously you are recognizing both A and B as self. And so you do not have, as part of your inheritance, any sort of antibodies here. And so you're not attacking any of the red blood cells. Okay? And then finally, we have type O. In the case of our type O, there is absolutely no antigen of this type hanging off of the cell membrane. So there's no A, there's no B, there's not an O antigen, it's just absent. Okay. Now in this case, that kind of clean red blood cell is considered self. So any A or B cells would be considered foreign. So as part of your genetic inheritance, you are inheriting and making these sort of large anti-A and anti-B antibodies. So you're recognizing A and B both as foreign. You are ready to immediately do an immune response against either of these types of cells. Okay, so again, just to kind of go through it one more time, if you are type A, you've inherited type A antigen and anti-B antibody, so you're going to attack any B marked red blood cells. If you're type B, it's the opposite. So you have type B antigen on your cells, you're making antibody anti-A. So you will, as part of your genetic inheritance, automatically, you've already got this antibody ready to attack any red blood cells with type A marker. If you're type AB, you have both of those antigens on your red blood cells, and so you're not making any of those antibodies. And if you're type O, it's kind of the opposite of type AB. This cell has no antigens, so it doesn't have A or B antigens, but it does make both anti-A and anti-B antibodies. Okay, so the real question then is looking at in blood donation, who can donate to who and what type of blood can you personally receive? So let's look at that next. I think I'll stick my camera view over here in the corner. All right. Well, maybe back up here. Okay. All right. So you're seeing over here in the far column, if you are type A blood, then you can inherit type A blood, obviously, but you can also inherit type O blood because it has no markers. As long as it doesn't have a B antigen on it, you're going to, it's going to be invisible to you. And then the same is true for for the type B. It's just the opposite. So obviously type B individuals can inherit type B blood, but you can also inherit type O blood because it's essentially invisible to you. If you are type AB, you can inherit any of the 4 types. Because you are seeing any red blood cell with an A marker as self, and you're seeing any red blood cell with a B marker as self. And because you don't have any of these antibodies, then you are not going to target any of these types of red blood cells for an immune response. If you're type O, you can only you can only receive type O blood because you're going to recognize A and B as foreign and you're going to attack those. Okay, so we can describe our type AB individual as the universal recipient, right? Because we've already said they can get anybody's blood. There's no problems. A and B are both self. So you can get just A, you can get just type B, you can get type AB, obviously. And then we've already talked about type O is essentially invisible to these people, to their immune response, right? In the case of type O, we call this the universal donor because as you see, because it does not have any of those antigen markers, it's essentially invisible to the immune system of type A, type B, and type AB people. And then obviously type O is only able to receive type O. So we see that it can be donated to anybody. The last bit of our discussion has to do with the Rh factor, and so this is what's described as positive and negative. A person who is positive means that they have the Rh factor as the antigen marker on the red blood cell. If you're Rh negative, then that means you don't have this marker on your red blood cell, okay? So again, I've been goofy, and I've written Rh hanging off of my cell. Now, the key thing to see that's different between our Rh factor and the type ABO is that in the case of Rh factor, you're not producing any antibody as part of that genetic inheritance. When we were looking a minute ago at type ABO blood type, we see that not only are you inheriting the information to make that antigen on the cells, but you're also inheriting the information to make that anti-A or anti-B antibody. In the case of the Rh factor, you are either inheriting the factor on your red blood cells or you're not, and that's the end of the story, sort of, okay? Now, if you are Rh negative, that means that that Rh factor is going to show up as a foreign antigen in your body. So if for some reason an Rh negative person becomes exposed to Rh positive blood, then an immune response would occur. Now we see this happening most often between mother and fetal blood supply. Okay, and so we see this discussion is important in what is called the hemolytic disease of the newborn. So hemo is, of course, blood, lytic is lysis or rupturing, or you can also call this erythroblastosis fetalis, and we'll see why it's called that name here as we go through. You're only going to see this particular condition showing up in cases where the mom is Rh negative and the fetus is Rh positive. So the only way that you can have an Rh positive fetus with an Rh negative mom is if the dad was contributing the genes for the Rh positive. And what we see is that sensitization can occur during that first pregnancy. The first pregnancy, you will not see the hemolytic disease of the newborn or erythroblastosis fetalis happening. You will only see this condition showing up with the second pregnancy or later pregnancies. Okay, so something to keep in mind is in pregnancy, of course, you have that really cool structure called the placenta. This is an organ that's actually shared by two people. So this is shared by the mom and the fetus. And then what we see is that you basically have the maternal blood supply and the fetal blood supply in close proximity to each other. So there's not actually any mixing of maternal and fetal blood. Everything is kept in those vessels. What we see, though, is that material will diffuse from the maternal capillary across to the fetal capillary, and then waste materials, of course, is going in the opposite direction. So during the pregnancy, there's no contact between the mom's red blood cells and the fetus red blood cells. And notice in this scenario that the mom's red blood cells are Rh negative, so they do not have that marker. And then I went ahead and used a little plus sign for my fetal red blood cells to indicate that they are Rh positive. Now, during the process of labor and delivery, there's lots of breaking of blood vessel that's happening in that placenta. So you're going to have rupturing of both fetal capillaries and maternal capillaries. So during this period, you can have some mixing of blood. So some of those fetal blood cells will actually move into the maternal blood supply. So now it's kind of like she is being infected by those Rh positive red blood cells. And so because those Rh positive markers are foreign markers, then her body is going to produce an immune response to those fetal red blood cells. So what that means is she's going to produce anti-Rh positive antibodies. And the signature feature of our immune responses is that we have memory. So the next time that she is exposed to Rh positive cells, her body can do an immune response against Rh positive really fast. And so we'll see that this will be the reason why we can see this syndrome of hemolytic disease of the newborn occurring in later pregnancies, but not the first pregnancy. So now we're looking at the second pregnancy. These antibodies are really small. Remember when I drew the anti-A and anti-B antibodies? These are fairly large structures. Let me make sure the pointer is on here so you can really see how big this is. When we're talking about those anti-Rh positive antibodies, it's just like that one Y shape, so significantly smaller. This small size of antibody will actually cross the placenta. So it will move from the maternal capillary into the fetal capillary. Now, this is good when we're talking about other antibodies because it is giving some immunity to that fetus so that when it is born, it's already having some protection from things that it'll be exposed to. But in this case, it's really bad because these antibodies are going to target the fetal red blood cells that are Rh positive. And what will happen is this will lead to attack and rupturing of these red blood cells. And so if your fetal red blood cells are destroyed, you're going to see that the fetus's cells in the bone marrow are going to increase the production of red blood cells. And so we're going to see that immature erythroblasts are going to be released. And if they're immature, they're not full functioning red blood cells, they don't have all that hemoglobin, they don't have all those properties of a mature red blood cell, they're not going to be able to move through the infant's cardiovascular system and all those tiny capillaries and deliver oxygen the way that they need to. So if this is occurring with the fetus, when that infant is born, that infant will require a complete blood replacement in order to survive. And so if that's not possible, then this infant is not surviving. So the erythroblastosis fetalis name is because since your red blood cells are getting destroyed, you're shooting out erythroblasts into circulation instead of mature red blood cells. So that's how we're getting that particular name. Okay, so what do we do about this? What happens if you're Rh negative mom and you're potentially carrying an Rh positive fetus? In the United States, all Rh negative pregnant women are given a shot of Rogaine. Rogaine is containing antibodies to destroy fetal red blood cells. So whether this is the first pregnancy, the second pregnancy, the third pregnancy, if you have Rh positive red blood cells entering the maternal bloodstream, she will attack and remove those rather than creating an immune response to those cells. So she will be removing the fetal red blood cells, but her body is not doing an immune response. She's not having memory. She's not making antibodies. She doesn't have any antibodies that can cross. And so her fetus is protected from her body, I guess we could say. This is an example of what we're going to call in chapter 22 artificially acquired passive immunity. Artificial because it's injected, acquired because obviously you're not born with this, and then the passive part means that her body is not producing an immune response. Those antibodies are being administered to her, and so then there's no memory associated with this sort of treatment. So every single pregnancy would require the Rogaine shot. All right, and that is it for now.